Students often ask how our muscles use energy sources when they are exercised. Okay, let's play. Let's say you have just stepped onto the treadmill at the gym. You start to run. There is a finite amount of ATP in your muscle cells. As you exercise, you quickly consume it. As you use the ATP already in the cell to cycle your myosin heads, there is an enzyme called creatine phosphatase that takes creatine phosphate and removes the phosphate group and transfers the phosphate group to ADP to recreate ATP. However, the activity level of this enzyme only lasts a few seconds. You are still running. Now what will you use to generate ATP? Next in line is the breakdown of glucose already in your muscle cell. Glucose can be anaerobically metabolized, which generates very little ATP, or aerobically metabolized. If you are running fast and not breathing well, your glucose stores will be metabolized anaerobically, and this means small amounts of ATP are generated and lactic acid will be created. The lactic acid causes your muscles to fatigue, and you will also feel discomfort or soreness as the levels build. You will ultimately stop running. However, if you are breathing well, the oxygen levels will allow you to metabolize glucose, fatty acids, and even amino acids delivered to your muscle cells via the bloodstream. Aerobic respiration means you are able to fully oxidize these metabolites and generate ATP. So, in summary, as you run, you use your creatine phosphate supplies first, then your muscle glucose and glycogen stores. These supplies can last for up to 20 to 30 minutes, and then you switch to fatty acid oxidation for ATP. In all, it takes about 30 to 40 minutes of exercise at about 40 to 80% intensity to start to burn fat. Sadness. These next two slides are really important. In your PhysioX Skeletal Muscle Physiology Lab, you will experiment with force summation. In other words, you will learn the ways muscle fibers can summate their contractions to generate a more forceful contraction. There are two ways of doing this. One, more motor units can be recruited. Remember, a motor unit is a single motor neuron and the individual muscle cells it innervates. A whole muscle has many motor units. Let's pretend you are picking up a piece of paper. How many motor units would contract in the biceps brachii? Now let's say you are going to pick up 10 bricks. Now how many motor units would be contracted? The answer is many more. This means that more muscle fibers are shortening, and overall the entire muscle is generating a more forceful contraction. The other way a whole muscle can generate a more vigorous contraction is if the muscle cell is stimulated repeatedly. With every stimulus, more calcium is dumped into the cytoplasm. More calcium means prolonged removal of tropomyosin's inhibition on actin, and more interaction of myosin with actin. More interaction or power stroking with actin means more contraction. It also means the calcium ATPase will take longer to remove the calcium from the cytosol to resequester it in the sarcoplasmic reticulum. Eventually, these contractions will lead to fused tetany. That means the whole muscle cannot shorten anymore. It has reached its maximum contraction. Let's go over the frequency summation again. In red, you see cytoplasmic calcium levels. In black, you see the force generated by the muscle. In blue, you see an action potential generated in the skeletal muscle. With every action potential, more calcium is dumped into the cytoplasm. More calcium means prolonged removal of tropomyosin's inhibition on actin and more interaction of myosin with actin. More interaction or power stroking with actin means more contraction. It also means the calcium ATPase will take longer to remove the calcium from the cytosol to resequester it in the sarcoplasmic reticulum. If another action potential arrives before the calcium has been resequestered, then that means even more calcium is now in the cytosol and more contraction results. Eventually, these contractions will lead to fused tetany. That means the whole muscle cannot shorten anymore. It has reached its maximum contraction. Students often ask how they can get more muscle mass. When you go to the gym, your increase in muscle mass occurs due to hypertrophy. This means that the number of myofibrils and myofilaments increases, so the girth of existing muscle cells increases. As we age, there is less opportunity for hyperplasia. This means that muscle cells undergo mitosis to generate new daughter cells. The immature cells that can divide to generate new muscle cells are called myoblasts. Again, as we age, the number of myoblasts decline. The evidence that endurance training can stimulate myoblasts to divide and generate new muscle cells is not robust, but there are some articles out there that describes this event. I had a friend in graduate school who was studying myoblast activity, and she was able to show that in young mice that suffered muscle injury, their myoblasts could generate new muscle tissue, but this declined as the mouse aged, due to a decline in myoblast cells. It's sad, but if you don't use it, you will lose it, at least when it comes to muscle mass. You can train at the gym and gain nice muscle tone, but if you stop training, your muscle mass will atrophy, which is the reverse of hypertrophy, through loss of myofibrils and myofilaments in as little as 2 weeks. If you really choose to live a sedentary lifestyle, the actual number of muscle fibers may decline, and you already learned from the previous slide how difficult it is to gain those fibers back through myoblast activity. The next few slides are a review of the central nervous system and peripheral nervous system. First, remember that the central nervous system is composed of only brain and spinal cord. Any nerve or neuron cell body found outside the brain or spinal cord is considered part of the peripheral nervous system. The spinal cord is really a, quote, go-between the brain and the peripheral nervous system. Its job is to conduct or relay sensory information from the body to the brain. Additionally, it sends messages from the brain to the body. What are these messages? How are they conveyed? Answer: through action potentials. In this segment, you will also learn about spinal reflexes. This basically means that sensory information from the body is quickly replied with an outgoing message or action potential that comes from the central nervous system. In this picture, you see a cross-section through the spinal cord. The blue axons in the picture are the axons that are attached to neuron cell bodies located in a dorsal root ganglion. These axons transmit action potentials toward the central nervous system and are therefore called afferent or sensory action potentials. The red axons in the picture come from neuron cell bodies in the ventral horn of the spinal cord gray matter. These carry action potentials away from the central nervous system to effector cells, and so they are called efferent or motor action potentials. Remember from anatomy that the effector cells of the body are smooth muscle, cardiac muscle, skeletal muscle, glands, and now we add on adipose tissue. In this picture, we see our efferent axon or fiber terminating on skeletal muscle. So this neuron is called a motor neuron. Now look at the right side of the picture. Notice where the blue and the red axons are bundled together in a nerve. Nerves are a collection of axons outside of the central nervous system. We say spinal nerves are, quote, mixed because they contain both afferent or sensory, and efferent, or motor, axons. What then would be the axons within the central nervous system shown here in purple? These bundles of axons within the central nervous system are called tracts. Find the structure labeled sensory ganglion on the right side of the screen. This is really called the dorsal root ganglion. A collection of cell bodies outside of the central nervous system is called a ganglion. A collection of cell bodies within the central nervous system is called a nucleus, or nuclei for plural. Is this material new to you? It shouldn't be. You should have learned about this in anatomy. If it is new to you, I recommend you read the nervous system supplements that are uploaded on Blackboard for you. Read them, and as always, come to your instructor during office hours for help. Now, let's review two important parts of the cerebral cortex. Remind yourself that the cerebral cortex is a thin outer layer of neurons. Within the cerebral cortex, we often find neurons that perform similar jobs. For example, shown here in blue is the precentral gyrus. The neurons found within this gyrus create the primary motor cortex. This means this part of the brain is responsible for generating the initial command for voluntary movement. In the brown area, you see it labeled as the postcentral gyrus. The neurons found here create the primary somatosensory cortex. The neurons here are important for interpreting action potentials into perceived stimuli. What do I mean by that? Pretend I have used a bone saw to cut off your calvaria, or the top of your skull. I place that in front of you on the table. Now I cut through your meninges, and your brain is now exposed to me. If I were to take an electrode and apply an electrical current to one area of your primary somatosensory cortex, I might stimulate some neurons there, and you would report to me the sensation of cold. If I touch another area, you may have the sensation of a paper cut on your right pinky. That reminds me to tell you that if I were stimulating your left primary somatosensory cortex, you would perceive a sensation on your right side of the body. That is what is meant by contralateral. Similarly, if I were to stimulate the right side of your primary motor cortex, you might all of a sudden pucker your lips. If I touch another spot, you might make a fist. The picture here of the primary motor cortex with pictures of the body drawn next to it, called a homunculus, it basically means that we have mapped the body parts that correspond to the neurons within the motor cortex. So, if I touch the lateral side of your primary motor cortex, your face would likely twitch or move in some way. This same type of body mapping, or homunculus, has also been done for the primary somatosensory cortex. The bigger the picture, the more neurons dedicated to serving that part of the body. For the primary somatosensory cortex, how big do you predict the picture for the tongue to be? What about your hand? What about your face? What about your back? In order to answer these questions, ask yourself how sensitive these body parts are. The more sensitive they are, the bigger the picture for that part of the homunculus. The spinal nerves are part of the peripheral nervous system, but it is important to remind you that you also have 12 cranial nerves. These nerves are also part of the peripheral nervous system. Cranial nerve 1 is the olfactory nerve and is sensory only for smell. Cranial nerve 2 is the optic nerve and is also sensory only for vision. Cranial nerve 3 is called the oculomotor nerve and is a mixed nerve. It is motor or efferent for 4 skeletal muscles that control eye movement: the medial rectus, inferior oblique, inferior rectus, and superior rectus. It also has involuntary effector fibers that control the intrinsic muscle near your iris, called the ciliary muscle in your eye. You will learn more about this nerve during your autonomic nervous system lecture, as it is part of the parasympathetic nervous system. Cranial nerve 4 is called the trochlear nerve, and it is a motor/efferent nerve and stimulates the superior oblique, another extrinsic eye muscle. Cranial nerve 5 is called the trigeminal nerve, and this is a mixed nerve. That means it has sensory fibers and motor fibers. Cranial nerve 5 allows you to sense pain or touch on your face and in your mouth and teeth, for example, and controls most muscles of mastication, your chewing muscles. Cranial nerve 6 is called the abducens nerve and is a motor nerve that controls movement of the extrinsic eye muscle called the lateral rectus. Cranial nerve 7 is called the facial nerve and is also mixed. Sensory for taste, motor for controlling your facial muscles, and the stapedius in the middle ear. No, you don't need to know the stapedius for your exam. And it is also the nerve responsible for releasing salivary secretions from the submandibular and sublingual glands, and also controls your tear release from your lacrimal gland. Cranial nerve 7 is also part of the parasympathetic nervous system. Cranial nerve 8 is purely sensory and is called the vestibulocochlear nerve and helps with transmitting information on sound waves and orientation of your body in your environment. In other words, your position. Cranial nerve 9 is called the glossopharyngeal nerve and is also a mixed nerve. It has motor fibers innervating some swallowing muscles in your throat. It is part of the parasympathetic nervous system. It has some involuntary efferent fibers that innervate your parotid gland, and this leads to more salivary secretions. And it also has a sensory component for taste. Cranial nerve 10 is called the vagus. It too is mixed. It has both sensory, motor, and involuntary motor fibers in this very important nerve. It will be talked about extensively during your autonomic nervous system lecture. Cranial nerve 10 is the only cranial nerve that extends from your brain all the way to your thoracic and abdominal cavities. Cranial nerve 11 is called the accessory nerve, and it is only somatic motor. It innervates your sternocleidomastoid muscle and trapezius. Lastly, the hypoglossal nerve is cranial nerve 12, and is motor only as it stimulates the muscles that move the tongue. Reflexes are rapid, automatic, involuntary reactions of muscles or glands to a stimulus. All reflexes have similar properties. 1. A stimulus is required to initiate a response to sensory input. 2. A rapid response requires that few neurons be involved and synaptic delay be minimal. 3. An automatic response occurs the same way every time. 4. An involuntary response requires no intent or pre-awareness of the reflex activity. Thus, reflexes are usually not suppressed. Awareness of the stimulus occurs after the reflex action has been completed, in time to correct or avoid a potentially dangerous situation. There are 5 components to a reflex arc shown at the top of this picture. 1. 1 shows you that a stimulus activates a receptor. Sensory receptors respond to external and internal stimuli, such as temperature, pressure, or tactile changes. 2 shows you a nerve impulse travels through sensory neurons to the central nervous system. Sensory neurons conduct impulses from the receptor into the spinal cord. And 3, you see integration. In other words, the information from the nerve impulse is processed in the integration center by interneurons within the central nervous system. More complex reflexes may use a number of interneurons within the central nervous system to integrate and process incoming sensory information and transmit information to the motor neuron. The simplest reflexes do not involve interneurons. Rather, the sensory neuron synapses directly on a motor neuron in the anterior gray horn of the spinal cord. In 4, a motor neuron transmits a nerve impulse to the effector. The motor neuron transmits a nerve impulse through the anterior route and spinal nerve to the peripheral effector organ. In 5, you see an effector responds to the nerve impulse from the motor neuron. An effector is a peripheral target organ that responds to the stimulus from the motor neuron. This response is intended to counteract or remove the original stimulus. Reflex arcs may be ipsilateral or contralateral. A reflex arc is termed ipsilateral when both the receptor and the effector organs of the reflex are on the same side of the spinal cord. For example, an ipsilateral effect occurs when the muscles in your left arm contract to pull your left hand away from a hot object. A reflex arc is contralateral when the sensory impulses from a receptor organ cross over through the spinal cord to activate effector organs in the opposite limb. For example, contralateral effect occurs when you step on a sharp object with your left foot and then contract the muscles in your right leg to maintain balance as you withdraw your left leg from the damaging object. Reflexes may also be monosynaptic or polysynaptic. A monosynaptic reflex is the simplest of all reflexes. The sensory axons synapse directly on the motor neurons, whose axons project to the effector. Interneurons are not involved in processing this reflex. Polysynaptic reflexes have more complex neural pathways that exhibit a number of synapses involving interneurons within the reflex arc. Because this reflex arc has more components, there is a more prolonged delay between the stimulus and response. Here are some reflex arc examples I am going to describe in the following slides. Monosynaptic stretch reflex, polysynaptic reflex arcs, which include withdrawal or reciprocal inhibition reflex, and the crossed extensor. The stretch reflex is a monosynaptic reflex that monitors and regulates skeletal muscle length. Stretch in a muscle is monitored by a stretch receptor called a muscle spindle fiber. It monitors passive tension. This means tension developed from the muscle being stretched. When a stimulus results in the stretching of a muscle, that muscle reflexively contracts. The patellar or knee-jerk reflex is a monosynaptic reflex. That is an example of a stretch reflex. The stimulus, or the tap on the patellar tendon, initiates the contraction of the quadriceps femoris muscle and leads to extension of the knee joint. If you did not learn about these in anatomy, you should come see me. Shown here is a picture of a muscle spindle found intertwined among the skeletal muscle cells. I also invite you to watch the video on the patellar reflex. What I want you to now know is that reflexes can be an important diagnostic tool. Clinicians use them to test specific muscle groups and specific spinal nerves or segments of the spinal cord. Although some variation in reflexes is normal, a consistently abnormal response might indicate damage to the nervous system or muscles. A reflex response may be normal, hypoactive, or hyperactive. The term hypoactive reflex means that a reflex response is diminished or absent. A hypoactive reflex may indicate damage to a segment of the spinal cord, or it may suggest muscle disease or damage to the neuromuscular junction. A hyperactive reflex refers to an abnormally strong response. It may indicate damage somewhere in either the brain or the spinal cord. A withdraw or flexor reflex is a polysynaptic reflex arc that is initiated by a painful stimulus, such as touching something very hot or someone forcefully grabbing your arm. Stimulation of a receptor organ causes the transmission of sensory information to the spinal cord. Interneurons receive the sensory information and stimulate motor neurons to direct flexor muscles to contract in response. Simultaneously, antagonistic muscles are inhibited so that the traumatized body part may be quickly withdrawn from the harmful stimulation. On the right side of the screen, you see the crossed extensor variety. Sometimes that painful stimulus elicits a flexor reflex in the affected limb and an extensor reflex in the opposite limb. This serves to push the body away from the stimulus or to shift the weight to the opposite limb. You can have 2 points of extra credit if you post on discussion board which neurotransmitter is released onto the somatic motor neurons shown on the left side of the spinal cord, where you see, quote, negative and the positive signs, and also which neurotransmitters are released onto the somatic motor neurons serving the extending forearm, where it says negative and positive. Hint: review your excitatory and inhibitory neurotransmitters that are used in the central nervous system. Post your answers on discussion board within Blackboard before your second lecture exam. The Golgi tendon reflex prevents skeletal muscles from tensing excessively during active tension. Golgi tendon organs are nerve endings that are located within tendons near a muscle-tendon junction, shown here. As a muscle contracts, its associated tendon stretches, resulting in increased tension in the tendon and activation of the Golgi tendon organ. Nerve impulses in the Golgi tendon organ signal interneurons in the spinal cord, which in turn inhibit the actions of the motor neurons. When the motor neurons are inhibited, the associated muscle is allowed to relax. This protects the muscle and tendon from excessive tension damage. This concludes our lecture. The only material that should be, quote, new to you should be slides 32 through 35 and slides 43 and 45. The rest, your anatomy instructor should have covered.